Hello, my name is Robert Brock. This is my honors thesis, the role of postsynaptic neuroligin adhesion proteins in neuron to OPC circuit formation. So from previous research, we know that neurons form synapses onto OPCs. OPCs are oligodendrocyte progenitor cells. This is a diagram of what that synapse looks like. So you have your presynaptic neuron here, synapsing onto the postsynaptic OPC. And in the synapse, we have this protein, Neuroligin, which has been shown to be important for forming and stabilizing synapses. And it binds to this other protein, Orexin. And so we don't know much about that synapse as to why it forms, but we do know that OPCs can differentiate into oligodendrocytes. Oligodendrocytes are the supporting glia in the brain that wrap neurons with myelin, which speeds up electrical conduction. And so we figured that these synapses may be important for myelination. Myelination, of course, is extremely important. Damage to the myelin sheath can cause disease phenotypes seen in multiple sclerosis, an autoimmune condition that targets the myelin. This causes an array of symptoms, and it affects about 2.8 million people worldwide. So to investigate this phenomenon, we took some animals, some mice, and we, on day 28, injected them with tamoxifen that activated this cascade of changes. For our experimental animals, it triggered this recombinase that excised the Neuroligin protein from the DNA sequence. And this stopped transcription and subsequently translation of the Neuroligin protein. And at day 35, animals were injected with an attenuated rabies virus that stained them red. And in order for this virus to work, it needed the activation of these other genes. The TVA gene allowed for the infection. The G glycoprotein gene allowed for it to spread monosynaptically. So this rabies virus spread from OPCs retrogradely across the synapse to neurons only one time, and also stained them red. And we would have had the activation of this reporter gene, this green fluorescent protein, but that ended up not working. So instead we went with a cellular staining method. We stained the OPCs in green and the neurons in this far red, sort of pink color. And so here are some representative images of what that looks like. You can see here the kind of yellowish cells, those are the OPCs in the corpus callosum. And here, these more red pinkish cells are neurons in the CA1 region of the hippocampus. And you can't see it at this sort of resolution, but you can imagine that there might be some processes here and a synapse forming between these two. And so here are all of the channel images. Here's the RAB-V, where you can see the red rabies virus, Olig2 for OPCs, Nlgn for neurons, and DAPI is a neuronal cellular marker. And so you weren't able to tell much of a difference between those representative images, but we do have some preliminary results which indicate a change. And so here on the screen, you can see each of these dots represents a different animal. And in the blue, you have the OPCs, in the red, you have the neurons, and these are the number of colocalizations, so the number of hits based on the different genotypes. And so we use this information to create a ratio of neurons to OPCs. And again, we organize it by group, the wild type, and Neuroligin 1 and 2. And I should note, Neuroligin 1 and 2 are different forms of the Neuroligin protein, so slightly different variations. And this is only preliminary data, but we did find a statistical significance here with the Neuroligin 1 group. in that they had a much lower ratio, meaning much less neurons were synapsing onto these OPCs. And so if we do find significant results, then this can inform what is necessary to build the synapse, which in turn gives us more information as to the nature of these OPCs and the synapses they form with neurons, and how it may impact myelination or the differentiation of these OPCs into oligodendrocytes. And this information is invaluable for developmental research, but also for disease research such as multiple sclerosis. Thank you.